Every dynamic, living process inside a mammalian cell, from respiration to replication, is entirely driven by the physical interactions of inanimate chemical structures. To understand biology, we have to follow a single, unbroken rule. Structure dictates function. That rule starts with water. Within its bent molecular structure, the oxygen atom attracts shared electrons more strongly than the hydrogen atoms do, a property known as electronegativity. This unequal electron distribution creates an electrical dipole, resulting in a partial negative charge on oxygen and partial positive charges on hydrogens. Because of these opposing charges, water acts as a highly effective solvent. Its polarity allows it to attract, pull apart, and dissolve other polar substances, establishing the fluid baseline of the cell. By establishing this aqueous baseline, water's polarity creates an inescapable chemical environment. Every other molecule introduced into the cell must either chemically integrate with the water or physically isolate away from it. Nonpolar molecules, specifically simple and conjugated lipids, lack these distinct electrical charges. Consequently, they cannot integrate with the polar water environment. When nonpolar lipids are surrounded by a polar solvent, a profound chemical repulsion occurs. The water molecules attract each other, effectively squeezing the lipids out of the solution. We can see why by looking at a lipid structure. It is amphipathic, possessing a hydrophilic or water-attracting head and a hydrophobic or water-repelling tail. When exposed to water, chemical repulsion forces lipids to spontaneously self-assemble. Hydrophobic tails hide inward, while hydrophilic heads face outward. This forms the lipid bilayer, the foundational boundary of the mammalian cell membrane, separating the inside from the outside world. In this cellular context, the physical properties of lipids provide structural containment. The mere chemical repulsion between water and lipids spontaneously engineers the physical walls of the cell. This isolates the internal environment, prepping it for complex biological machinery. With the boundary established, we can examine the next layer of cellular architecture, proteins. These large molecules embed themselves directly into the lipid boundary and float within the aqueous interior. Proteins function as the active machinery of the cell, performing highly specific mechanical and chemical work. A protein's ability to do this work relies on how its raw chain of amino acids physically bends. This linear chain is the primary structure, which quickly begins folding into secondary structures, like helices and pleated sheets. This animation tracks the sequence as those shapes fold further into complex three-dimensional tertiary structures and eventually lock together into quaternary structures. When a protein achieves a specific tertiary fold that allows it to accelerate a chemical reaction, we classify it as an enzyme. Mammalian digestive enzymes, like pepsin, are prime examples of these highly specialized shapes. These enzymes are biological catalysts. Without them, the metabolic processes required to sustain a living organism would occur too slowly to support life. An enzyme does not function because of some invisible life force. It functions because its exact three-dimensional folded structure allows it to grip and manipulate other specific molecules. How does a precisely folded enzyme physically grip its target molecule, which we call the substrate? Historically, biologists relied on the lock and key hypothesis. This idea assumed the enzyme was a perfectly rigid structure, and the substrate fit into it exactly, like a key sliding into a metal lock. But biological evidence showed that enzymes are flexible. Mammalian metabolism is highly dynamic, and a perfectly rigid structure could not explain how enzymes physically catalyze reactions and release the resulting products. This split screen compares the older model with the currently accepted scientific model, the induced fit hypothesis.
On the right, the enzyme and substrate are not a perfect match initially. When the substrate enters, the enzyme physically shifts its structural shape to firmly grasp it. This conformational change in the enzyme puts physical stress on the chemical bonds of the substrate, lowering the activation energy required to trigger a chemical reaction. The enzyme chemically converts the substrate into a new final product and then immediately releases it back into the cellular fluid. Once the product is released, the enzyme snaps back to its original resting structure. It remains completely unchanged by the reaction, ready to repeat the process indefinitely. This dynamic flexibility is highly specific. An enzyme will only induce a fit and undergo a conformational change for its one designated substrate. Mammalian enzymes operate as dynamic resetting machines. Their structural flexibility is the physical mechanism that drives cellular metabolism. To prove this mechanical relationship, scientists can measure and manipulate the speed of these enzymes in a laboratory setting. This study is called enzyme kinetics. By tracking the rate of a chemical reaction against the concentration of available substrates, we can see exactly how structural availability limits biological speed. This graph plots the reaction rate on the y-axis against substrate concentration on the x-axis in an environment with a fixed number of enzymes. As we add substrate, the rate climbs. We see a steep, linear rise in the reaction rate. Because there are many empty enzyme active sites, the substrates easily find them, bind, and react. Eventually, the system reaches a point of saturation. This occurs when the substrate concentration is so high that every available enzyme is simultaneously occupied. Notice how the graph line plateaus completely. Because all the structural machinery is currently in use, adding more substrate to the environment has zero effect on the reaction speed. The only way to raise this plateau and increase the maximum rate of reaction is to physically add more enzymes to the baseline environment. Biological reaction rates are strictly limited by physical enzyme availability. Metabolic speed is ultimately a structural numbers game. Just as a cell needs to catalyze reactions, it also needs to slow or halt them to maintain balance. This process is known as enzyme inhibition. This diagram illustrates two methods of stopping an enzyme. The top shows competitive inhibition. An imposter structurally mimics the real substrate and enters the active site. By occupying that space, it physically blocks the actual substrate, stopping the reaction. The bottom shows non-competitive inhibition. Here, a molecule binds to a different location on the enzyme. This binding forces a structural change throughout the entire protein, warping the active site and destroying its specificity. The substrate simply bounces off. Whether an inhibitor plugs the active site directly or warps it from afar, the rule holds true. Slightly altering an enzyme structure modifies or terminates its biological function. These microscopic mechanisms of inhibition directly inform modern macroscopic medicine. Medical researchers design pharmaceutical drugs specifically to act as targeted enzyme inhibitors. Many common antibiotics and pain relievers work by physically blocking or warping the enzymes that bacteria and pain pathways use to function. Commercial industries use these exact same principles of structural manipulation to control large-scale biochemical processes, such as managing enzyme kinetics in commercial fermentation. From the simple polarity of water, to the assembly of lipid boundaries, to the precise folding of proteins, the chain of causation is clear. Every macroscopic biological outcome is dictated by the precise physical interactions of chemical structures. The study of this molecular architecture allows medical researchers to design precise interventions that inhibit disease and stabilize cellular health.